If an average person started reading it, it would take them 9 years to read one person's book of life.
It was in this very pub that Francis Crick and James Watson proclaimed to have found the secret of life. They discovered the structure of DNA. Since then, great leaps have been made in the sequencing and analysis of DNA, a field better known as genomics. In 2003, the first ever human genome was created. Genomics has come a long way, most famously with the Human Genome Project, which, whilst groundbreaking, may have a representation problem. 80% of all insights come from people of European descent, which means that there are millions of gene variations still missing. I'm Dr. Shini Somara, and I've come to Cambridge to meet a company that's hoping to diversify genomics by travelling further afield to collect data. Deoxyribonucleic acid, or DNA, is found in all organisms. It's made up of 4 chemical bases known as nucleotides. They are adenine A, guanine G, cytosine C, and thymine T. DNA bases pair up with each other. A goes with T and C goes with G, forming what's known as base pairs. There are 3 billion base pairs arranged along 2 spiralling strands, known as a double helix. Each of us has around 20 to 25,000 genes, which encode all of the characteristics that make us who we are. Genes vary in size from a few hundred DNA bases to more than 2 million. We know this thanks to the Human Genome Project. In 2003, scientists created the first ever genome sequence based on DNA samples from volunteers. This became the standard against which all other DNA sequences were compared. But it had one major drawback. 70% of the reference genome was based on the genes of just one man, while the rest came from about 50 other volunteers. Every person's genetic code is unique, so using just one reference genome, most of it from the genetics of one man, to stand in for humanity has introduced subtle biases into genetic research. Sumit Jamwar is the head of Global Gene Corp, which is spearheading efforts to diversify genomics by collecting biodata from India. He's based on the Wellcome Genome campus, where the first ever human genome was sequenced.
This captures the magic.
The human genome.
Absolutely.
These 112 books contain the DNA sequence of the entire human genome, essentially what is the genetic blueprint for building a human being. So initially, the idea with the Human Genome Project was to create one reference human genome. And the work that you're trying to do is really drill down into the detail of the variations of that code contained in this set of books.
That was the hypothesis. That was the reason why people went in with it and assumed that if once we have the reference genome, humans only have a small variation, so it should all work for us. And what we have realized over time is that each one of us is different in a slight but a very significant way. So if you were to read, what you see is basically a series of letters A, T, C, and G's arranged in a different way, which cuts across in a sequential manner.
So much information, and this is just one book.
If an average person started reading it, it would take them 9 years to read one person's book of life. On an average, there are 3 million points where an individual is different from another person. And while that may not seem like a lot, it's actually very significant in terms of driving and understanding and the risk that we are exposed to and putting that across, right? So you're absolutely correct. It started with a handful of volunteers, and what we now need to do to achieve the promise of genomics is to understand what does a good look like for different populations that exist, because it helps us understand what the risks are for that particular individual from that particular population, but it also allows us to solve for different diseases on a global scale.
What's going on in here with these huge machines? I mean, it looks so impressive.
And they are. I mean, what you see here is these are some of the latest machines which are being used to sequence the DNA. And what you see there, if you look at the lines there, the blue lines that are going across, that means that the sequencing is in progress. This site is one of the largest sequencing labs that exist in terms of throughput. But those machines are the latest machines that you can see, which are creating the DNA sequences to progress science that happens out here.
Julia Wilson is an Associate Director of the Wellcome Sanger Institute, which was a key player in sequencing that first reference genome. So it took 13 years to get to the end of the project. Mission accomplished. Why do you need this institute and what are you doing now?
Well, that was just the first. That was just one single human genome. We didn't even know we didn't even know how many genes there was until we'd done this. We knew there was 3 million bases, that's the letters of code, but we didn't know that there was 20,000 genes, what they did, how they work together, and what the differences are between individuals, some with disease, some without disease. And so this institute now sequences thousands and thousands of genomes to look for those patterns that could possibly remain obscure.
Is it a real problem that the Human Genome Project is based on essentially one man and then a handful of other volunteers?
Well, yes, and we have to start somewhere, but now we see efforts to sequence different and more diverse populations. So there's efforts in Asia, in Africa, and so it's important to be able to contextualise against a reference genome for those populations.
Is there a massive difference between your genes and my genes?
No, we're about 99.99% similar. Even though we're so similar, it's those small spelling changes that make the differences.
Global Gene Corp are hoping to achieve genomic diversity by collecting DNA samples from across India. The man bringing together the data to achieve this genomic diversity for Global Gene Corp is Will McLaren, a chief bioinformatician.
So India is particularly interesting. So in our research we've found that there is around 4,500 different ethnicities in India, which makes up a significant proportion of the ethnicities worldwide. So we see up here this pink and red area represents the South Asians. So the red ones are the South Asians as part of the 1,000 Genomes Project, and the pink ones you see here are samples from our study.
So if we were to just be focusing on what the Human Genome Project tells us, we would only be looking at this green blob here.
Exactly, and there's all of this unexplored space outside of the green area here, which should become part of the global genome.
So as a bioinformatician, what's your dream for the future of this research?
I would like to see making 70 the new 40. So we want people who reach the age of 70 to be in the same state of health that they are now at the age of 40 through this kind of advancement in genomics and science.
So much science in that story. Don't forget to like, comment and subscribe and hit the bell button below for notifications.